I'm going to assume that people can hear me on the phone line, and again, we really apologize. Do you want to get the please? So if you could go to the next one. Maybe I need to repeat just quickly that the slides are attached in the handout. So if you would like to follow along, if you're having difficulty seeing, or if you're on the phone line and you would like to follow along, the slides were sent to you in an email. So look for those and you can follow along with the webinar today. And there are some requirements listed here. You do need to attend the live webinar and be registered or be in a room with someone who is. If you're on the phone line, just go ahead and send your name to this email address and we'll make sure we get you a certificate. Okay, next slide, please. So I'm going to go ahead and move forward and introduce our first presenter, Greg Sales. Greg is the director of the Center for Environmental Solutions and Emergency Response in EPA's Office of Research and Development. Greg, I'll go ahead and turn it over to you.
Just got started a couple weeks ago, and they're really just starting to generate some data now and get ready to start generating data. But we're really excited to share with you, sort of where we're headed. EPA does already have robust capabilities to respond to environmental emergencies, and in particular biological contamination. And the whole agency is doing that right now. However, in addition, this COVID virus is, of course, a new experience for all of us. And so we are turning some of our expertise in EPA's Office of Research and Development to try to, deliver useful tools to folks around the country, public health agencies, states, locals, etc. So our work is aimed at really enhancing the capabilities of states and territories, tribes, local governments, so that you all can help advise the public, businesses, and institutions better. We're partnering a lot within the agency, but also with external entities such as CDC, several state short-term and all research questions that were here. They include, how can we best disinfect real-world surfaces? How can we disinfect people so we can keep reusing it? What do we do about high-touch public spaces that are perhaps infected every few minutes? And how do we deal with that? Are there long-term disinfectants? Are our wastewater treatment plant, the raw sewage coming into them, are they good indicators of community infection? And finally, are there analytical methods that we've been really good at developing in EPA that could be adapted to this problem and help the nation? So the next slide, please. So what we're going to do is we're going to have a series of speakers who are going to give the introductions to their body of work that they are shepherding. So the first one is Dr. Sean Ryan. He's going to be talking about the projects related to cleanup and disinfection. Sean has over 15 years experience of doing research in EPA, especially related to Homeland Security and disinfection and decontamination. and he is an EPA.
EPA, and that's important to understand. EPA has over 420 products that are currently approved for use against COVID-19. The vast majority of these, over 400 of them, are for use on hard, non-porous materials such as countertops or hard plastics or metal or glass surfaces. There's only a handful that really are intended for use on soft lawn for frequently used porous materials. These products that are used on hard, non-porous surfaces such as metal and glass, how well do they work on surfaces that are kind of in between hard and non-porous and soft and porous materials? Materials like rubber on escalator handrails or latex painted surfaces. And what are some of these non-laundering options for things like seat fabric, like you might find in subway rail cars, some subway rail cars, or some buses, or airplanes? And so this research project, we're working to try to provide those answers. We're looking at products that are currently on EPA's list for use against SARS-CoV-2, also called List N frequently. We're looking at products that are typically or are used against hard, non-porous materials and testing their applicability against some of these more commonly encountered materials. The goal is to be able to provide reliable data on the efficacy of available products and then work. So with an understanding of which products might be appropriate to address these different disinfection needs, the next question that really comes to mind is how do you appropriately apply them? And it's very important to say here that you need to make sure you follow label instructions, that you use appropriate personal protective equipment, such as gloves or anything else that might be listed on the label, that you apply the products appropriately, and that you ensure that surfaces remain wet for the entire specified contact time that might be on the label. Many products, such as those that you see every day in the stores, you'll see them come in like spray bottles or what they call trigger spray type mechanisms. So if you can think about having to apply that over a pretty large area, maybe like an entire rail car or an entire wall type area, you get pretty tired, squirting the trigger mechanism. So for these larger applications, there's really an interest others or compatible with the different types of disinfectant that are currently on the market, especially ones that are listed more for those trigger or tested as trigger sprays. So can the effective application parameters, such as keeping the surface wet for the entire contact time, can that still be achieved when you do use a fine mist or a fog? Are the active ingredients that are in these disinfectants, are they impacted by these different application methods? And then are there any additional safety concerns using these kind of application methods with these disinfectants? And these are the types of questions that we're addressing in this study to expand our understanding, to understand the effectiveness and safe use of these type of application methods for common disinfectants. Next slide, please. So there are many, sir, we should be especially product stain resistance or odor pieces. They can typically prevent the growth of odor or causing bacteria or mold or mildew. But these products aren't registered for use as disinfectants or providing long-term virucidal properties. But wouldn't it be great if they were? We could provide resistant properties over days to weeks to months. And so that's the purpose of this testing that we're looking at. We're looking at the efficacy of different surface protectants and how well they do as disinfectants against SARS-CoV-2, the virus that causes COVID-19. How well do they work over time, a week, a month or so after their application to a surface? And how well do they hold up to normal cleaning or normal wear due to the normal use of these kind of surfaces with the coatings on? Results, of course, are of high interest to mass transit operators as well as other stakeholders that have many high-touch surfaces in public areas. Next slide, please. We should be slide 11. So in addition to these chemical products that we talked about on the last couple of slides, there's also a high interest in the use of alternative methods such as UV, ozone, and steam. UV and ozone are routinely used in water disinfection, and UV is often used in the healthcare setting. Those provide unique uses of where UV and ozone really do work well. But the question is, let's get them out of those settings, and we need to use UV or underneath a handrail in a subway car. So the question is, do these technologies still work, or can they still be deployed to work in these kind of situations? And that's what this effort is looking at, addressing this for both UV, for ozone, what concentrations of ozone work well, and then for steam, what is the contact time and of the steam on different surfaces as options for disinfecting, especially porous materials. Next slide, please. So as many of you have probably heard, personal protective equipment, including masks, gloves, gowns, face shields, are in high demand. And many of these items really are meant for single use. However, now, because of these shortages, they're being used multiple times. And so the questions that come to mind are not a great number in everyday scenarios. So that's the purpose of this effort. We're looking at different types of PPE. We're looking at different methods that may be suitable and available for disinfection, seeing how well they work as disinfectants and also how well the different PPE can stand up to disinfection. Next slide, please. We should be on slide 13 on surface sampling efficacy. So surface sampling in the environment for viable virus is highly variable, and it's not overly informative. There are challenges with efficiently collecting viruses from surfaces. There's challenges with getting the virus out of sampling media, such as a swab or a wipe that's used to sample a surface. And there are challenges with the analysis. Sampling for pathogens from different surfaces, it's not very sensitive and can have a very high detection limit, meaning that not detecting a virus on a surface does not necessarily mean that surface is clean. It also requires several days or a week for analysis to confirm whether or not the virus is still infective. In the meantime, the virus is naturally decaying on surfaces. So the results that we have now seven days later really do not tell us what is currently on that surface in terms of viral load. So there's that challenge as well as many of the others that is reporting viral RNA do not indicate that what the actual potential exposure risk is to the infected virus. So for these reasons and many others, disinfectants are routinely used without confirmatory sampling. And it's really felt that the stringency of the disinfectant test methods and proving that a disinfectant works really provides a high degree of certainty that when a disinfectant is used as directed, it's going to be effective. And adding additional surface sampling because of the uncertainty of helpful to support epidemiological studies, for example, assessing how well containment practices are working, or if activities are leading to the spread of the virus outside of certain areas, for example, outside a patient's room. So for that reason, understanding of the information that can be determined from it for SARS-CoV-2, we're evaluating sampling collection efficiency and limits to detection for SARS-CoV-2 on common surfaces and by using common sampling techniques for biologics. We should be on slide 14, which is COVID-2 rapid molecular viability assay. So as mentioned, and on that previous slide on surface sampling, the analysis of environmental samples for viruses can also be problematic. Methods that detect viral RNA, for example, they do not directly inform on the actual infectivity of the virus. RNA detection methods, they can be rapid, they can be done within a few hours or a day if you consider the sample processing to analysis, such as looking for any human coronavirus. However, they do not indicate that, they indicate the RNA is detected, but they don't tell you if that virus is still infected. So that determination really requires infectivity analysis, which can and added several days to complete that analysis, and then additional analysis to confirm that whatever just killed the cells in that infectivity analysis are actually the SARS-CoV-2 virus. So it's a pretty complicated process. And we're really trying to improve this in our work now by combining the infectivity piece and the selectivity of the RNA detection piece through PCR into what's called rapid viability reverse transcriptase polymerase chain reaction, or RVRT-PCR. It's, again, a technique that can combine those two analysis methods and really hopefully reduce the time for sample analysis. This is work that's being done in collaboration with the Department of Energy and Lawrence Livermore National Labs. It's building upon work that EPA has done developing RV-PCR to support environmental sampling for Bacillus anthracis spores, and the objective here is to reduce our sampling time. And for that, I'd like to introduce Dr. Kevin Oshima. Dr. Oshima is a division director in EPA's Office of Research and Development, and he uses his degree in environmental virology and strong research background to lead research supporting public health water microbiology needs. Dr. Oshima?
Sean, can you hear me?
I can hear you, yes.
Awesome, thanks. So we're going to switch gears now to talk about COVID-2 and sewage, so the development of standardized methods. The challenge, so COVID-2 has been detected in fecal specimens from patients diagnosed with COVID-19, as well as raw sewage. So that's for the precipitation of this interest. GI illness is a common symptom found among COVID-19 patients. There's increasing evidence of detection of virus in patient stool samples, as well as limited detection to it. And it also illustrates the need for well-characterized methods in order to be able to interpret the results and also to widely disseminate methods across the country. So, why do we want to look at what's the kind of information that we can get from looking at COVID-2 loads in sewage? Well, one is it gives us a better sense of what the treatment process and the levels of virus that is found in raw sewage. So from a public health perspective, but also since sewage is a collection of whatever the human population is contributing towards it, and if COVID-2 densities in sewage is readily measurable, then perhaps it can be used as a way to assess community infection within populations. So for those reasons, we are interested in developing COVID-2 detection methods in sewage. So sensitive, standardized methods are needed for detecting and quantifying COVID-2 in raw sewage, including infectious virus. The concentrations in, so one of the aspects of it is, in order to work with concentrating sewage samples and also looking at infectivity, you need to utilize more methodologies within the laboratory. So CDC, for example, currently recommends BSL-2+ conditions for concentrating raw sewage. If your methods that you're using requires the concentration of raw sewage samples. And if you culture, then that requires a BSL-3 level laboratory safety. To date, most of the data that's been collected has been using molecular methods. That in this case, this is an RNA virus. There is also the need to test for the infectivity of the virus in raw sewage, and that typically utilizes culture methods. So that's another aspect of what needs to be done. Based on other studies with coronaviruses in general tend to be not very stable in aquatic settings. Nonetheless, this will be a way to confirm that. There's development and application of these methods. Initially, we're going to be working with surrogate coronaviruses. These are human isolated from humans, but require BSL level 2 rather than the more infectious and pathogenic COVID-2 virus. From there, we're going to be looking at detecting COVID-2 directly from these samples, and we'll be using BSL-3 conditions for both concentration and for concentration of the virus from sewage, and also for culturing the virus from these samples. We'll characterize the performance of the methods by spiking the surrogate viruses. We're looking for recovery efficiency and reproducibility. So we'll be developing a standardized method and validatable and demonstrating the performance. Quantifying the level of COVID-2, once we've got a method, we'll be then applying that method or methodologies to field samples to look at the level of COVID-2 and by both molecular and live virus measurement in raw sewage from wastewater treatment plants. And again, it depends on what we find in raw sewage. The deliverables will be delivering validated standardized methods for concentrating and detecting COVID-2 in raw sewage using molecular infectivity measurements. We'll be reporting documentation of COVID-2 levels in raw sewage using that standardized method or methods, and then report on the effectiveness of wastewater treatment on COVID-2. For scientists, I'm just going to talk about the application of sewage in terms of community assessment.
Thank you, Kevin. Can you hear me?
Yes.
Great. So if we go to the next slide. What I want to talk about today is very closely related to what Kevin was discussing about the impact of SARS-CoV-2 in wastewater. And in this case, it's not necessarily a challenge, it's an opportunity, because the early evidence suggests that the levels of RNA associated with SARS-CoV-2 can be potentially a leading indicator of rising infection levels in the population. Thought to be because there's so many significant number of asymptomatic individuals that as you have relatively low infection levels of documented infection, there's a number level or community swap as opposed to individual testing. To replace individual based testing where you can understand who's infected and who's not, and you can then isolate the people that are sick, but it may allow you to target large groups of people initially to help focus your subsequent either your targeted testing and hopefully inform community public health decisions. So that's the opportunity. The challenge is that there's complexity to this. In addition to what Kevin was mentioning about having consistent methods that you can apply to wastewater with all the proper controls, so that when you get a measurement from wastewater, you're confident that it's not a variation occurring in your methodology, in the fact that it is reflecting what's going on in the community. So that's one, understanding the dynamics in the sewer itself, understanding the residence time and decay rates of that viral particle in the systems, stormwater, rainwater, into the sewers. It could be also industrial discharges. So that's another component of the work that needs to be done. And then finally, you're trying to relate the temporal changes in your sewer signal back to dynamics in infection levels in the community, the donating community in this case. So it's complex, and it requires both the understanding of the analytical part, the application of that analytical part to sewage, a complex media, and as well as the understanding the engineering of the sewer systems, and also interfacing with public health officials. So the work that I'm going to talk about and our work moving forward involves close cooperation, as Kevin mentioned, also the CDC. So our approach, I think as Kevin mentioned, there's very active work going on in developing and validating a method. And I think in a perfect world, we would focus on developing that validated method, and then we would apply it to monitor the sewer. But we now know that, we don't live in a perfect world. It's very dynamic. So we are trying to do these efforts in parallel fashion. So as we develop a more validated method, we want to apply a self-consistent method with relatively acceptable recovery rates. And so that's what we're doing first. So we say rapidly assess with the molecular approach. That's really, we're focused on over the next 4 to 6 weeks, really based on the available literature, based on the available knowledge, really rapidly develop the method and apply it to sewage to really begin to understand how it works within this complex medium. And we're working closely with our colleagues here locally in Cincinnati, the Cincinnati Sewer District, as well as other utilities predominantly across the state of Ohio that we've linked up with over the last month or so. After this initial period of really the method assessment, we're going to then try to develop a longer-term pilot monitoring effort or shift the targets of the sampling based on dynamics in the pandemic. This initial pilot monitoring effort, we're going to continue to work closely with our colleagues at MSD, not only because they very much understand the dynamics of their system, they model the flow, they know the residence time in major treatment plants here in the Cincinnati area. And that allows us to really evaluate some important factors that could be influencing the signal, including we have combined and non-combined sewer systems here in Cincinnati. The major one is combined, but we also have some smaller ones that are non-combined, therefore they don't have that rainfall input. And we can begin to look at ways to the impact of dilution and ways to mitigate that dilution. For example, instead of expressing the genome copies per liter of wastewater, in addition to that, we can use other markers for fecal content and understand the impact of different normalization approaches on the stability of the sewer signal. We also want to work with CDC, I mean not CDC, but MSC in Cincinnati. There's a number of industrially active degradation of the signal because of the nature of those wastewaters. We also have reached out, like mentioned, across the state of Ohio to work with various utilities, and including some of relatively hot spots of infection, particularly related to correctional institutes in the state of Ohio that are either have their own independent wastewater treatment plant, as in Pickaway County, Ohio, or linked to an existing city wastewater treatment plant, as in Marion County, Ohio. And those will allow us to they'll help in the initial method evaluation too, because there'll be a high signal. In addition, that allows you to kind of have a very targeted community, a very well-characterized community, because one of the reasons why we have high reported infection rates in those areas is that there's been extensive testing at the individual level done in those prisons. So in terms of deliverables, one of the things we're really trying to focus on is not only develop data for publication and for kind of scientific interpretation, but we really want to develop a pilot monitoring effort where we're turning the data around within a week time frame, so that before we go out and sample the next week, we already have processed the data and made it available to both the utilities and the relevant public health agencies, so they can actually be used as part of their assessment protocol. The state of Ohio, the governor just this week has reached out to ourselves and universities across the Ohio, because he very much wants to implement a statewide surveillance program using wastewater. So we're beginning to work with the other potential partners, in this case the utility, other university labs, so that we can develop a network of surveillance that may not involve a single standard method that everybody's using, but adopting minimum data requirements for everybody, so that we can compare the data, and that we really can have a statewide surveillance network. This is something that CDC, in our collaboration with them, they're very much interested in doing that as well at a national level, kind of having a national repository of data, but also building in this minimum data requirement idea to accept data into this national data kind of repository. We're trying to use this Ohio kind of as a pilot for doing some of that in collaboration with CDC. CDC also has developed a method or a model linking the sewer signal all the way back to community infection. And we're working with them on targeting some of our studies here to best inform both the development and validation, some sets of population, useful to help validate that model. For example, if you have a very well-characterized community, like in a prison, and you're monitoring the level in the sewage directly from that, you can really kind of, like I said, validate and evaluate their model. And we also then hope to have obviously a report kind of summarizing lessons learned and recommendations for sewer, for the plant, but also within the network, into certain targets within the network where we can look at the sources of potentially high-risk populations, and focus in on sources and communities that may not be influenced by, say, industrial discharges, and then compare the signal within the sewer network to the overall integrated sample. So I'm going to end there, and I'm going to turn it over to Tim Wade, who's our ORG's lead epidemiologist. He's going to talk about some work on exposure.
Okay, great. Jay, can you hear me?
Yes, I can.
Okay, good. Great. Okay, thank you. Can go ahead and advance on to the next slide, please. So, yeah, good afternoon, everyone. And today I'll be discussing the development of a salivary antibody assay to detect recent infections with SARS-CoV-2. So as most of you probably know, the widely used diagnostic test for SARS-CoV-2 infection is a PCR test that detects the virus's genetic material in nasal or oral swabs. So because it detects the presence of the virus, it only detects the presence of the virus. It can tell you if you have a current infection, but cannot determine whether an individual has been previously or recently infected and recovered from infection. Antibody tests, on the other hand, measure the presence of immunoglobulins or antibodies specific to a particular pathogen. Usually, specific antibodies rise steeply following an acute infection as part of immune response. Antibodies to SARS-CoV-2 usually appear from 1 to 4 weeks following infection, and they may remain detectable for a variable period of time following infection, which we don't really know yet, but from months to possibly years. So antibody tests can be used to identify previously infected individuals, even those who may have been asymptomatic, and better track the extent of the infection in the population. When antibodies develop and how long they can be detected depends on many factors, such as the nature and intensity of the infection, the type of antibody, immunoglobulin M, A, or G, and differences in individual immune responses. Most standard antibody tests for COVID-19 and other infections are done using serum and require a blood and saliva has the advantage of being simple and painless to collect, and collection is well tolerated even by children. Also, it can be self-collected and returned through mail without special preservatives or buffers. Salivary antibody tests have been developed for other pathogens and other infections. One well-known example is the screening test used for HIV infection, which can be done at home outside of clinical settings and produce results in about 20 minutes. Another early example of saliva testing for population infection rates was in the UK about 20 years ago, when it was used to assess population infection rates of hepatitis A, and was done simply by mailing collection kits to study participants across the country. Our group at EPA began working on developing non-invasive salivary antibody tests for waterborne and other infections about 15 years ago, and developed research assays for a variety of waterborne, soil-borne, and community infections, including several noroviruses, one of the most common. This custom assay has been successfully applied in prospective studies of drinking and recreational water-associated infections, as well as population-based community studies. We developed these using a multiplex Luminex platform, which allows us to test the antibody responses to multiple antigens, pathogen targets that trigger a specific immune response, as well as internal controls in a single test. These multiplexing capabilities can greatly reduce analysis costs and the necessary volume of saliva. We've had particular success developing an assay for norovirus, which is a virus that has several different strains and can vary from year to year. And we were able to validate this assay using samples from known infected individuals and applied it in population-based surveys across the United States and demonstrated linkages with risk factors as well as asymptomatic rates of infection. The examples of our saliva collection was shown on the previous slide, and on the slide currently shown. The collection device is called an Oracol sampler, and it consists of a simple sponge attached to a handle. Collection involves rubbing the gums gently for about 1 minute until the sponge is saturated and returned to the collection tube. This technique focuses on the collection of crevicular fluid, which is usually more enriched in antibodies and tends to mirror antibodies in serum. Over the years, we've collected over 20,000 saliva samples using this kit in a wide range of communities and settings. And we're also experimenting with a passive drool collection method that is also non-invasive. So developing a salivary antibody assay for SARS-CoV-2 will build on this experience. The main challenge is identifying antigens that produce the strongest antibody response, validating the assay using samples from individuals with confirmed infection versus uninfected controls. Interpretation of antibody results can also be challenging due to individual variation and the timing and intensity of the antibody response. Saliva samples have some other challenges, including sample-to-sample variability in their composition, especially when people are self-collecting them, and lower levels of some antibody types compared to serum. Despite these challenges, a non-invasive salivary antibody test for SARS-CoV-2 has the potential to enable low-cost population surveillance of surveillance status identifiers for infection and severe illness, and especially potentially looking at disproportionately affected populations. We would like to emphasize that this tool, we are developing it, is for research purposes to better understand the COVID-19 infection and its population dynamics, and not for clinical diagnostics. We are at the early stages of this assay in development and are currently working to obtain positive control samples, both serum and saliva, from previously infected individuals, working with other researchers, medical clinics, and biobanks to obtain these samples. For negative controls, we can rely on our repository of serum and saliva samples from previously conducted studies. We'll also be starting our own study, which will involve recruiting recently infected individuals and collecting saliva samples through the mail. And we are obtaining and testing a range of possible antigens and testing. And I'll conclude by acknowledging the lead researchers and the rest of the team that has been working hard on this effort. And I will turn it back to Greg, I guess, or I'm not sure who I'm turning it back to. Thank you, everyone.
Thank you, Tim. This is Greg. Can you hear me, Tim?
Yes, I can.
Good. All right. Well, we're going to wrap things up now, but let me just give you a little bit of information. So I hope you've enjoyed this introduction to a body of work. Very exciting work that we're doing in EPA. We've really pivoted to redirect a lot of effort towards the work that we really want to get out, get started and get out to the nation over the coming months. To stay in touch with this work, there's a number of ways. We have a brand new website, which is outlined there on the page. If you follow that, then you'll follow our updates. We will have periodic webinars as well that you'll hear the announcements for. We'll have fact sheets coming out over time, and our intent is really to give not only final reports when we're done with all this work, but interim results as much as we can, so that states and communities around the nation can use the work. Let me go to the last slide here and just thank the speakers today. These are all sort of amazing researchers who have really started to run with this work and are sprinting, and we're really trying to get things cranking and get results out as soon as possible. At the bottom there is Lonnie Mattis-Curry, our communications director, who is also instrumental in getting our word out today. So thank you, everybody. I'm going to hand it over to Michelle for questions.
Thanks, Greg. Can you hear me okay now?
I can.
Okay, great. Before I move forward, we're going to have we do have 5 minutes, so we're going to actually try to get as many questions in as possible before the top of the hour. I do want to thank all the presenters and all of you as well, all of you on the line, all of you logged into the webinar today. I know there were a lot of technical issues up front, and we really apologize for that. But we will, as you can see here on the slide, we will have this webinar posted online, so you can go back and watch it. And I know there's a lot of questions that have come in. We can't get to all of them today, but we will follow up. We do have your contact information and any questions that you've asked. So we will follow up with you, and as Greg said, there's contact information here if you have follow-up questions for the presenters or Lonnie as well. I'm going to go ahead and start back with Sean. So I'm going to go back to the beginning of the presentation. and ask each of the presenters one question since we only have a few minutes. So Sean, and this was on cleanup and disinfection evaluation. Sorry, my main question disappeared here for a second, so I may have to go ahead and change that to a different presenter. Or Sean, actually, here's a question for you. On using ozone for room disinfection, do you have an idea of when basic guidelines might be available? For example, size of output of the ozone generator for an X number of cubic feet of room?
So this is research that we are just starting up with the SARS-CoV-2 work, so it's going to take a little bit of time. So I would guess sometime early fall, I would hope that we would have that kind of specific information out for ozone. We do have some information for other agents that can help to give kind of a ballpark, other biological pathogens that could help to give a ballpark and can give a good estimate now, but in terms of specific data, it would probably be in the fall.
Okay, thanks, Sean. Kevin, I'm going to move on to you on wastewater. There were a couple questions on this. Is there any evidence of people becoming infected with the virus who work at wastewater treatment plants, or any evidence of people becoming infected with the virus through contact with wastewater?
Thanks. No, we're not aware of any data out there that suggests there's a linkage between. Of course, this is a rapidly developing disease, and so there's new information coming in. But other than the detection part of it, I'm not aware of any direct evidence of transmission via that route.
Okay, thanks, Kevin. Jay, this question's for you, also about wastewater and monitoring. Are you working with any other groups outside of EPA, such as Stanford, the groups that may be already monitoring wastewater treatment plants?
We're definitely communicating with a number of different people, and that's an important point. There's a number of universities, also private companies, doing monitoring. So I think one of the challenges is like I said, trying to develop, if not the exact methods, but the minimum data requirements needed to compare across methods. So yeah, we are aware of that, and we've participated in the WRF, the Water Research Foundation Summit on this topic. So we're aware of all the work that's going on, and trying to build networks for monitoring at a broad scale.
Great, thanks, Jay. And Tim, we're going to go ahead and end with you with a question for you on the assays. And the question is and you may have talked about this a little already, but is it available in just the U.S. or will it also be available in Canada, maybe other countries as well?
Our intention for this is to make it available for research. So we actually intend to publish it and not use it as a diagnostic approach. But having said that, absolutely, I think that the methods and the techniques will be, the goal would be to make them broadly available, and that would either be through publications or other kinds of public dissemination of our work and how we did it.
All right, great. Thanks, Tim. All right, well, everyone, thanks again for joining us. We will let you know when this recording is available. Keep hold of your slides and use those links. The recording should be available within a week or two. And if you do need a copy of the slides, you have any trouble with certificates, anything, just go ahead and email. There's an email address on the second slide of the webinar today. All right, thank you all for joining us, and we hope to see you at our next webinar.